We have talked through ways that the gene pool can be reduced, but now let's talk about two ways that they can be expanded. The first is through sexual reproduction, or meiosis. Mitosis is asexual reproduction and results in clones being formed. The genetic material of the daughter is exactly the same as the genetic material of the parent. However, in meiosis, we start with a diploid individual, so 2N. In meiosis 1, we duplicate those to get a tetrad. So there's a brief time where we have 4N before we divide it up again back to 2N. However, one of the things that's important to note is that if we had crossing over, because those tails of the chromosomes intersected, now our 2N looks different from the 2N that we started with. Still diploid, but because of crossing over, we have some more variation. And then the last step, because this all happened in meiosis 1, in meiosis 2, we will get a 1N, or haploid, gamete, where this cell will divide, and this is the chromosome that I'm going to take. This is meiosis 2. And if you need a review of meiosis, I will link both of my previous meiosis videos in the description below. The real bonus here in expanding our gene pool, not only do we have a gamete from one parent, the same thing is happening in the second parent. And maybe crossing over left a little bit of a different pattern. And the pattern is going to be different because they didn't start off exactly the same. These are two distinct parents. I'll say that this is parent 1 and this is parent 2. And so when we combine these into a zygote, that zygote is going to be genetically unique and have a different combination of genes and alleles than either of the two parents. So in terms of a gene pool, you could start with the population of red triangles. My triangles are getting messier and messier as we go on, but that's okay. And blue triangles. And then over time, our population changes so that we still have the blue triangles and we still have some red triangles. But maybe this was a mom and this was a dad triangle, and the offspring that resulted didn't look like either of the two parents. So I'm going to just say, for this example, that the offspring were purple triangles. Their genetic identity is different from that of both parents. This allows us to expand our gene pool. There's one more mechanism that can result in an expansion of the gene pool that we're going to talk about, and that is gene flow. One way that I remember gene flow is as the opposite of the founder effect. So with gene flow, instead of starting with one population and splitting and moving into two populations, we're going to start with two populations that are separate. Population 1 is going to have just red triangles. Population 2 just has green triangles. For gene flow to occur, something must happen to allow for one of these individuals from one population to relocate to the second population. After you get that movement, I'm going to redraw population 2. Population 1 is going to look the same, just going to have one less red triangle. But population 2 has green triangles, just as they did before. They didn't move anywhere. But now has a red triangle. And over time, if we allow these triangles to reproduce, we'll get more of the green, but we'll also get more of the red. So here, we are increasing the genetic diversity of the population, specifically population number 2, and as a result, we've expanded our gene pool.